and the scientific projects and education team, I would like to welcome everyone to this informal webinar that we will focus on our Discovery Quest program of awards. This actually is the second of the series of webinars that we have implemented in our team to provide resources to apply to CERN funding. The information contained in these slides and webinar presentations and the Q&A will be made available through our website, along with FAQs derived from our interactions with applicants and derived from today's Q&A as well. Just as a reference, this is being recorded, so it will also be made available afterwards. So if you need to leave in the middle of the webinar, just don't worry, we will have it available for you afterwards. Our hope is that these webinars become a resource for current and future applicants and that help answer many of the questions that we encounter. The goal could be that the applicants for this program consult with our team and our staff only when you need to discuss very specifics of your research project and how does it fit into our programs. Before going into the agenda for today's webinar, I want to take the opportunity to introduce our scientific and programs and education team. Some of you are familiar with some of the faces, but we have two new members of the team, so I'm going to go over who we are. Most of you are familiar with Kelly Shepard. Kelly, Associate Director in our team, has been with CERN for the past 13 years, working on different programs and advising many of you on how to develop successful applications for those funding programs. Ute Grishammer, she joined us back in June of 2021. As you know, Ute was at CERN from 2007 to 2015, and she led programs that created both our genomics initiatives and our iPS cell bank. She also organized many conferences and workshops involving hundreds of CERN-funded researchers. After leaving CERN, Ute became the scientific director of the California Initiative to Advance Precision Medicine at UCSF, and she created and managed applications and peer review process there. She joined us back in 2021, and she's been leading the Shared Resource Labs program, RFA. So we are actually going to pause very soon these RFAs. And for those interested, the concept was approved back in October, so you can check our website for the details. And she's also been very involved in the implementation of our data sharing and management plan guidelines to the Discovery programs. Joining Kelly, Ute, and I are two new members to our team that we are very excited to introduce to you today. Chan Lek Tan, he goes by Chan, has a PhD in molecular and cellular neuroscience from the Rockefeller University, where he studied under Dr. Paul Greengard. He carried out postdoctoral work in the laboratory of Zachary Knight at UCSF, where he worked in central circuits controlling thermoregulation. He brings also to us industry experience in pre-IND development of a small molecule and RNA-based therapeutics for neuro-based indications. As you all know, we have $1.5 billion earmarked for diseases of the brain, and his expertise in neurodevelopmental and neurodegenerative diseases aligns well with some of those priorities. Janie Bydom joins us as a science officer. She has a PhD in biomedical sciences with a concentration in immunology from the University of New Mexico Health Sciences Center in Albuquerque, and a postdoctoral training in immunology from Columbia University. She brings deep cell biology knowledge, and her most recent role was as an R&D engineer at the Chan Zuckerberg BioHub, where, amongst other things, she built imaging pipeline projects to interrogate the impacts of viral infection on cell function. Her breadth of expertise ranges from molecular immunology and cell engineering to high-throughput screening and advanced microscopy. With this background, you will know now who your science officers are and a little bit more about them. We only have an hour for this, so I'm going to go into the agenda for today. we have divided, thank you, Doug. We have divided today's agenda in two parts. The first one is an overview that will last 40 minutes at the maximum of CERN mission, strategic plan, and pillars of funding, which is going to be me, and it's going to be just 2 more slides. And then we are going to move into the Quest Awards. This is what we are going to cover, and the outcomes, the expected outcomes and eligible candidate types, the scope and activities, applicant eligibility requirements, review process and criteria, application components, and how can you find them, resource for applicants, common questions and mistakes. And in the second part, we are going to go through question and answer. The next slide is our mission statement. You are all very familiar with it. So I'm going to move into the next slide, please. Over the past 15 years, the way that we've been achieving this mission has been through the coordination of a very well-established model of based on these 5 pillars of funding. As you know, we have infrastructure, education, discovery, translational, and clinical. Our team actually handles the education program, which right now we have 4 different programs, all the discovery, and then infrastructure. And we can talk about some part of the infrastructure, sorry. In order to advance the mission, this plan is organized in 3 main strategic themes. And the one that we are going to talk about today is framed within the first strategic theme, which is advancing world-class science. The main goal of the advanced world-class science is to leverage collective scientific knowledge to inspire collaborative research. And some of the changes that you have seen in the program announcements and in the applications and the requirements that we have to do with the current alignments of programs to the newest strategy. We are actually working towards a shift in the way that we think and fund research and how it can be aligned in a collaborative manner to maximize the leverage of our funding. My colleagues are actually now going to go through the next slides and provide an overview of the specifics of how we are thinking about these programs, and then we will answer any questions that you have. As a reminder, this is being recorded. So Kelly, you want to just take over from here?
Thank you very much, Rosa. Now, before we get into the details of the DIS2 program, I want to let you know that we have a lot to cover, and we're going to do our best to finish on time. But if it takes us a few minutes extra to run over, we will answer your questions. And as Rosa just mentioned, this is being recorded, and we will make an effort to answer questions and provide the responses to you after the recording and webinar output are available as well. So let me just begin by introducing the slide to you, which depicts the core funding opportunities that CERN offers. We call these our R&D pillar programs because they're a series of funding opportunities that support research at different stages, beginning from the earliest stages of discovery, when you're exploring new ideas and testing hypotheses, all the way through the clinical trial stage, ultimately leading to an approved therapy or perhaps commercialization of a technology. So the focus of our team and of the program that we're going to be discussing today is in the discovery pillar over on the earlier side of the pipeline, as you see here on the left. The DIS0 or Foundation Awards was the topic of a webinar that we offered last fall and is available to you if you're interested in that program. We're going to focus on our other discovery program, the DIS2 or Quest Awards, which has been around for a few years now, since 2016 or so. And this is an important program because this program is something that is going to lead to a candidate that there is an expectation can come onto this pipeline, and the product will ultimately evolve through a series of developmental stages to where it can become a commercialized or clinical product. So I'm presenting this slide because I want you to have this vision in your mind, because when you put together a DIS2 proposal, you should be envisioning where this product could go if it's successful, either through your own subsequent development or through handing it off or passing the baton to our partner eventually. Next slide, please. Thank you. So before I plunge into DIS2, I just want to spend just a minute differentiating the two programs a little bit. programs a little bit because we do receive a lot of questions. So while both support early stage discovery, they're very distinct in what the expectations are when you put together a proposal. So as I mentioned previously, the DISC Zero or Foundation Awards we discussed previously in a webinar. This is our earliest stage program. It's really about addressing knowledge gaps in the field of regenerative medicine. It supports basic exploratory research. And there is no expectation that the outcome of a foundation award would necessarily progress into the development path. However, this knowledge can act as a foundation that could potentially lead to development candidates through further funding, or could potentially help other laboratories and other investigators throughout the world develop new ideas or technologies that can advance regenerative medicine. Now, on the other hand, the Quest Awards or DISC-2, which we're talking about today, there is a very specific outcome that is expected, and that is a development candidate that will be ready to move into translation by the end of your DISC-2 award. There are two types or tracks that are supported through our current DISC-2 funding opportunity. The first is the therapeutic track, where the expected outcome or goal of your program would be to identify a therapeutic candidate that would be ready to move into translation by the end of the award. This track offers up to $1.5 million in direct project costs, and I'm emphasizing direct project costs because that does not include any indirects or facility costs that might be applicable to a given application. The duration of support for this program is 36 months. And if successfully developed, an eligible therapeutic candidate would be expected to meet readiness to come back and apply for subsequent funding, potentially through a Tran 1 award or perhaps another funding opportunity. Now, the other track for our DISC-2 program can be lumped into what is considered a technology candidate. Technology candidate is a candidate that is not going to go down the therapeutic path. It could be a tool, it could be a device candidate, it could be a diagnostic test candidate. There are different varieties of tools or technologies that this could comprise. The support level for this path is $500,000, again, direct project costs. The duration is 24 months. And if successful, a prototype or development candidate that emerges from this program could potentially be competitive for funding under a subsequent opportunity at CIRM through our translational stage program. Next slide, please. Next slide. Thank you. So this is a high-level overview of what we're going to be focusing on for the next part of my presentation. So I just explained what the expected outcomes were for the DISC-2 program, which would be a therapy candidate or a technology candidate. But this expected outcome comes with some deliverables, and these are clearly defined in the program announcement. Briefly, these are the elements that we think are important to have as deliverables for these outcomes in order to ensure that you will meet the readiness to enter early development or translation. So for therapy candidates, that means throughout your proposal and by the culmination of the activities that you propose, you should have developed or identified a single candidate that will be ready for translation. You will have demonstrated reproducible disease or injury modifying activity for that candidate. You will have proposed or identified some measures of identity, activity, and purity. And you will have worked on a draft target product profile to accompany that development candidate as it makes its way through the R&D pipeline. Similarly, for technology candidates, the goal of this program is to produce a prototype tool or technology that will be ready for translation by the end of your funding period, demonstration of the performance of that tool or technology in a test model that's relevant to its intended use, demonstration of technical feasibility to meet design requirements, and again, a draft target product profile that can accompany your development candidate as it progresses through later stages of development. Next slide, please. Thank you. So let's delve a little bit deeper into therapeutic candidates. What types of therapeutic candidates are eligible for support under DISC-2? These also fall into a couple of categories. The first category I want to cover with you are cell therapies. So only specific cell therapies are eligible under DISC-2. These are cell therapies where a human stem or progenitor cell composes the therapy, or where human stem or progenitor cells are used to manufacture the therapy. By this, we mean, for example, through a differentiation process where the cell therapy is a differentiated derivative of a stem cell. Now, stem cell-based therapies can be gene modified, or they could include potentially other components, making them what would be considered likely a combination product down the road. DIS2 also supports genetic therapies, and there's a definition for how CIRM defines gene therapy under this initiative in the program announcement, but basically it's a therapeutic that acts by altering the genome of a cell or manipulating nucleic acids in cells. To be qualified for DIS2, a potential genetic therapy should target a human somatic cell for its therapeutic effect, and it should be regenerative. In other words, it should be intended to replace, regenerate, or repair the function of aged, disease-damaged, or defective cells, tissues, and/or organs. There are several other therapeutic modalities that can be supported under DIS2. However, there are some limitations around them. To be eligible, small molecular biologic candidates, for example, they should have an activity that depends on a stem cell, basically. So a small molecular biologic that acts on an endogenous human stem cell for its therapeutic effect, it should potentially act or target a human cancer stem cell for its therapeutic effect, or it could perhaps modify a stem cell therapy. Other examples of eligible therapeutics include those where a human stem cell is necessary to manufacture the therapy. For example, a stem cell-derived exosome would be considered a biologic, or where human stem cells are uniquely required for candidate identification and testing. Next slide, please. On our tool track, there are several different types of tools or technologies that would be eligible, including, as I mentioned, potentially diagnostic types of candidates, or those that might fall under a device or tool category. And these kind of fall into two major categories of eligibility. The first applies to tools or technologies that are actually made from stem cells or include stem cell components. These are human stem or progenitor cell-based, for example, test devices or technologies that can be used to discover things or advance, monitor, or evaluate new therapies. The other category are for tools or technologies that are not cell-based, but they could be, for example, a piece of hardware or an imaging modality or something along these lines. In order for those types of tools or technologies to be eligible, they should be rationalized as addressing a critical bottleneck, either to the discovery, development, or use of stem cell-based therapies, or to the discovery, development, or use of genetic therapies. In addition, for this category, you do need to propose testing that includes stem cells, human stem cells, if you are addressing a stem cell bottleneck, or any relevant human somatic cell type or stem cell type if you're targeting a gene therapy bottleneck. Okay, next slide, please. Okay. Now that we've talked about the eligible type of candidates, I want to briefly go over what types of activities are in scope. So basically, DIS2 supports activities that contribute to the selection of a novel candidate that can immediately progress into translation to enable broad use, as I described. So activities that will help you meet this expected outcome. Most importantly, you should design a proposal with activities that will lead you or culminate in a demonstration of proof of concept. I'm going to spend a few minutes going over proof of concept in more detail in my next few slides, so hold on to that if you have questions about that. Other eligible activities here include developing and implementing assays to identify, test, and characterize the candidate, assessing feasibility and reproducibility of your proposed candidate, developing the target product profile, and in some cases, if relevant, it may even include preparation for and conduct of a stage-appropriate regulatory meeting, and that might be applicable in some of our more mature but still early discovery stage programs where an interact meeting with the FDA could potentially be in scope if the project was at the appropriate stage. You'll notice that a lot of these activities that I mentioned are the ones that will lead you to the deliverables that will put you in a good position to be ready for translation by the end of your discovery award. Next slide, please. So let's just spend a few minutes talking about what we mean by proof of concept, because an important part of achieving the expected outcome of this DIS2 is demonstrating proof of concept for the candidate that you're working to discover. So for stem cell-based therapy candidates, with or without gene modification or other components. Some examples of a stem cell-based therapy would be pluripotent stem cell-derived cells such as neurons or islets, any PSC derivative, gene-modified hematopoietic stem cells, primary human neural stem cells with biomaterials, etc. So for this category, proof of concept means demonstrating reproducible disease modifying or injury modifying activity with the candidate that you intend to be translated in a preclinical model that is relevant to your target indication. Now, a couple of considerations you need to make if you're proposing a cell therapy candidate. Is it going to be an allogeneic approach or an autologous? If you are proposing an allogeneic candidate, you must design your experiment so that you will show your final proof of concept using a product made from the same cell line or cell source that's intended for translation. And this should be, in the case of any allogeneic cell, it should be a line that you will be allowed to take into a clinical trial, meaning it should be clinically compatible, meaning it should meet the FDA's donor eligibility requirements. If, on the other hand, you are proposing to develop an autologous cell therapy candidate, you're going to want to design your proof of concept studies to show evidence of disease-modifying activity using product made from 2 or more donor sources in order to establish the reproducibility of the process. This is really important to show readiness for translation, that it doesn't work with just the one cell line you tried, it works on multiple. Okay, next slide, please. For genetic therapy candidates that are not also stem cell therapies, we have a slightly different definition of proof of concept. So an example of this type of genetic therapy candidate would be in vivo delivered genes, for example, through viral vectors, antisense oligonucleotides, mRNA-based therapeutics, etc. This category also covers ex vivo gene-modified somatic cell therapies, so non-stem cell-based cell therapies that are gene-modified, for example, CAR T cells. So proof of concept here is demonstration of reproducible disease or injury modifying activity with the candidate to be translated using a clinically relevant model. And this is very important. You must also include evidence that the genetic therapy candidate targets or has activity on a clinically relevant human cell population. So if you don't already have evidence that the candidate will work on a human cell, much as it will on an animal cell, which might be what you're showing in your disease-modifying activity experiment, you should include an additional study to show that this candidate can in fact target or have some activity on a human cell. Now, there may be some rare exceptions where this is not technically possible. If you think that could apply to you, please contact members of the scientific programs team to discuss before starting your application or before submitting it so that we can discuss this with you. You'll receive contact information at the end of the slide for how to do that. Now, one point I want to make before we move on is that if your genetic therapy candidate is composed of cells, for example, a CAR T cell type product, then the allogeneic autologous consideration for proof of concept applies to you, as mentioned on my previous slide. Okay, next, please. Thank you. So now we will talk about what we mean by proof of concept for drugs or biologics candidates, things like small molecules, antibodies, peptides, exosomes. The definition here is reproducible disease-modifying or injury-modifying activity with the candidate to be translated on or with a clinically relevant human stem or progenitor or cancer stem cell population, whichever is your target of your therapy. This is critical to have this human-based evidence in order to meet readiness for our translational stage programs. Now, a couple of caveats to mention here. If you are developing a small molecule candidate, if this small molecule is going to require further optimization that would take longer than the DIS2 project period, for example, if you need to do medicinal chemistry or structure modifications to improve the drug properties, that candidate will not be ready for translation at the end of the ward, and that would not be considered an eligible project. So just take this into consideration that if you're proposing a small molecule study, and if you think you're going to need to do lead optimization, be sure to incorporate those studies and make sure they're feasible on the timeline of the 36-month period for a therapeutic candidate. That is your goal. And finally, if your biologics candidate is itself manufactured from a human stem or progenitor cell, for example, MSC-derived exosomes as an example, disease-modifying activity can be demonstrated without the extra requirement for testing on or with human stem or progenitor cells. Next slide, please. Finally, I just want to spend a minute going over what defines proof of concept for the technology type candidates. Examples are things like stem cell-based disease models for drug screening as a platform, diagnostics that incorporate stem cell components, or tools for use with stem cell or genetic therapies. Just a couple of examples. So the definition here is demonstration that the prototype, which would be your candidate that you're developing, meets initial performance criteria and a test model relevant to the intended use. So for any tool or technology, you would be defining the intended use. For example, a diagnostic candidate would be intended to distinguish a disease state from a healthy state. So your proof of concept would be to demonstrate that your prototype can detect the difference between a disease state and a healthy state. Now, for that category of tools and technologies that do not include stem cells, or they're not made from stem cells or from cells, your proof of concept will also be to demonstrate utility of that tool as described above. However, you also do need to propose testing with the cells that your device or tool is intended to be used with. So if your project is addressing a stem cell bottleneck, you should be employing at some level human stem cells in the testing to show your proof of concept. If, on the other hand, you're proposing to address a gene therapy bottleneck, then you could be using any type of human cell that you argue is relevant to the bottleneck that you're addressing to test the utility of your candidate. Just a little footnote that if you are developing a tool or technology that includes cells, such as allogeneic cells, you'll need to make sure that those cells have been appropriately consented for commercial development at some point in order for that tool to be considered for translation in subsequent funding opportunities by CIRM. Okay, and finally, I just want to go over what activities are not supported by DIS2. Primarily activities that are seen as too premature or too mature. So translational and clinical stage activities that are supported by our development programs are definitely not supported in DIS2. Things like developing GMP compatible processes, GLP studies, preparing for and conducting IND filings, clinical trials, etc. Also, any activities that are too exploratory and have not been justified as necessary for achieving the expected outcome of DIS2. And there is one set of activities that are conditional, and I haven't talked about the special supplement fund yet, and we're going to get to that a little bit later, but there is a special supplemental funding line item in the budget available to a certain class of projects, those that are developing allogeneic human pluripotent stem cell derived cell therapies. There is a special supplement available to acquire and use those lines in your testing for the purposes of candidate selection. You're not allowed to use that supplemental funding to work with or purchase lines that do not meet the definition of clinically compatible. And again, we'll go back over that a little bit later in this presentation. Next slide, please. So now let's talk a little bit about who's eligible to apply for a DIS2. So first of all, only California-based institutions can apply. A principal investigator on an application must commit at least 20% effort to this project. PI is limited to one DIS2 application per review cycle. A PI must not have a substantially similar application with overlapping activities pending review under any CIRM funding opportunity. Your award must be able to start within 90 days of the CIRM board's funding decision or approval of funding. And a special case for for-profit applicants only, they must demonstrate solvency to be eligible to apply. Next slide, please. Thank you. Now I'm just going to take a few minutes to go over the review process. Thank you. So this slide is depicting the standard review process for CIRM. So if you've ever submitted an application to CIRM in the past, you may be familiar with this. Once the application has been submitted, it will undergo an eligibility screen to make sure that the submitted application meets the basic eligibility requirements, including the ones that I went over with you today. Once an application has been deemed eligible, it would be forwarded to the grants working group for review. The grants working group are a panel of scientists from outside of California. And there are also some patient advocate members of CIRM's governing board that participate on the grants working group. So applications that are reviewed are scored and critiques are provided by the scientific members of the grants working group. Ultimately, the entire grants working group makes recommendations, which go to our board, the ICOC, specifically the application review subcommittee, in order to make final decisions for funding. Next slide, please. I do want to point out that for our discovery stage programs, they are highly subscribed. We receive large numbers of applications that have to date always exceeded the capacity of our grants working group to review in a single review cycle. So when that happens, we implement an extra step in the standard review process. It breaks the review process down into 2 steps. The first step is called positive selection. It occurs between the time of the eligibility screen and the scientific scoring. So let me go over very briefly what positive selection looks like. Next slide, please. So again, for high volume programs, the review process takes place in 2 stages for eligible applications. The first stage is called positive selection. And during this process, the grants working group members, that includes both the scientific members as well as the patient advocate members from CIRM's board, they're going to conduct a pre-review of all applications. No matter how many they are, they're going to conduct a pre-review of all of them, and they're going to identify a subset that they believe are the most impactful and responsive. There's a special section of the application that they're going to look at first to help them make this decision called the positive selection preview page. You're going to hear a little bit more about that later. To move forward to the second stage of review, applications that are selected by any given grants working group member in the first stage will move to stage 2, which is the CIRM standard review process. Applications that are moved to stage 2 are discussed by the entire group. They receive scores and critiques and feedback. Applications that are not selected to move forward to stage 2 do not advance, nor do they receive critiques, but they may be considered for reapplying in the future. Next slide, please. So what are grants working group members looking for when they score applications? These are the review criteria by which they're making their assessments. Does the project hold the necessary significance and potential for impact? Is the rationale sound? Is the project well planned and designed? Is it feasible? And importantly as well, does the project uphold principles of diversity, equity, and inclusion? Next slide, please. Okay, so that concludes the part of my presentation. I'm going to hand things over to my colleague, Janie, to go over the components and considerations when you're actually starting to put together an application.
Thanks, Kelly. Next slide, please. So once you've created a login for the application on the grants management system, there's a sidebar that shows all the components of the application that you can see in this graphic. And the online components will indicate their completion status to the right. And the upload section down at the bottom contains the required templates for things like the research proposal, biosketches, and a document where the PI lists other funding support that they have. Next slide, please. And so as I showed in the previous slide, the online components include filling out details about your budget, key personnel, et cetera, and a summary page where you can sell the key points of your project for high volume review. And this is called the positive selection preview page. The components that we'll complete offline and upload in the upload section are the proposal, other support, biosketches, and for for-profit applications, the financial feasibility and solvency forms. This is also where you can upload optional non-templated documents like letters of support. Next slide, please. And these are just example images of the templates. Again, these are found in the upload section, and for each template, there are page limits that are in each section of the proposal. And so that's where you'd find the page limits are in the templates themselves. Next slide, please. And so for-profit applicants have additional required uploads, and the templates for the financial feasibility assessment form and the financial solvency spreadsheet are also included in the upload section. Next slide. So CIRM has a responsibility to track data and metadata to promote fair data sharing principles, meaning that the data generated in CIRM-funded projects should be findable, accessible, interoperable, and reusable. And applicants can submit a data sharing overview as part of their proposal in the application, and detailed instructions with examples are provided in the proposal template. Generally, applicants will describe the types of experiments and the data generated, how the data is going to be processed, where the raw data, metadata, methods, and analyzed data will be deposited as well. And so after the grant has been awarded, the grantees will submit a data sharing and management plan, or a DSMP, and this contains more details for cataloging the data, metadata, and any tools developed for analysis. If you've submitted a previous DISC application in a recent cycle, you may remember that a separate DSMP template was provided for completion at the time of application. So we've changed that requirement so that at the time of application, you'll instead provide this data sharing overview, and then later upon funding, you'd submit a DSMP. Though applicants are not submitting the DSMP with the application, it's still a good idea to familiarize yourself with the DSMP template so that you understand the expectations if your award is funded. Next slide, please. And a few notes on the proposal component itself. Please use the required Word template and adhere to the prescribed page limits that are listed in the template. Maintain the formatting, like the fonts and the margins. Follow the instructions that are provided with each section. And then when you're about to upload the proposal, convert it to a PDF. Again, don't alter the margins or the font size, and please adhere to the page limits. And please do not use your own template. Next slide, please. So when you're resubmitting an application, if you're resubmitting an application that was not funded in an initial submission and you were not scored in the positive selection, meaning you did not receive comments from review, when you can submit your application as a new application. And if your initial application was discussed by the grants working group and you received a merit score, you can select yes to designate as a resubmission in the information for review section, and you would complete a resubmission statement. And the resubmission statement is really your opportunity to respond to previous reviewer comments and highlight any improvements or updates that you've made to your application. When resubmitting, we recommend downloading the latest template from the upload section, as the template sections can change from time to time. Next slide, please. And I'm going to pass this off to Ute for some resources that you can have while you are applying to the DISC awards.
Thanks, Janie. Doug, if you could just go back one slide. I just wanted to give you a brief outline of the 3 resources that I'm going to cover. I'm going to talk, and you've heard already about 2 of them, briefly tell you about resources relevant to clinically compatible human pluripotent stem cell lines, about the data sharing and management plans, and also about your opportunity or ability to look at previous review summaries. Next slide. I just wanted to help you navigate the CIRM website. When you land on CIRM's homepage, in the top bar, you can see, if you click on for researchers, you'll find various resources there. And right now, the 2, click again, Doug, please. If you click on information for applicants, that's where you will find more details around clinically compatible human pluripotent stem cell lines and DSMPs. Next slide. Kelly already mentioned the requirement that DISC-2 grantees who develop an allogeneic cell-based therapeutic candidate, that they must employ a cell line that meets CIRM's definition of clinically compatible. And so before I go into the great resources we have for you regarding this, I wanted to take an opportunity to emphasize a little more what do we mean by clinically compatible. There are 2 items that need to be true for a cell line to be clinically compatible. The first is that it meets the good tissue practices requirements for donor eligibility. And what that means is that the original cell donor whose cells are transformed and used, not transformed, used in developing a cell product, must have undergone a check for certain communicable diseases, must be free from those diseases. That's the GTP requirement. I also want to take this opportunity to emphasize that this is not the same as GMP, which Kelly already mentioned. Good manufacturing practices, development of reproducible and controlled production of your cell product that you can work on in a TRAN award, for instance. That's not a DISC-2 activity, but the cells have to be meeting GTP requirements, and the cells that were donated for an allogeneic product have to be appropriately consented by the cell donor. I also want to emphasize that while CIRM has an iPS cell repository that contains thousands of cell lines, they're all for research use only, and they do not meet this definition of clinically compatible. Kelly already also mentioned, importantly, that we do have a special additional budget that you're eligible to apply for if you are, for the purpose of candidate selection, to evaluate multiple clinically compatible human pluripotent stem cell lines. Doug, if you could click twice. I'm highlighting here this extra budget that you might be eligible for is only in cases where you're developing an allogeneic human pluripotent stem cell-based therapy. Next slide. Showing you again where you find on the CIRM website these resources. What we do have is in this section is an opportunity for you to view information on providers that are known to CIRM, providers of clinically compatible human pluripotent stem cell lines. These providers, and some of these providers, as you can see when you navigate there and study this table in some more detail, some of these providers make their lines available to CIRM awardees as part of the CIRM Industry Resource Partner Program. So you can, there's contact information if you're interested in acquiring some of these lines, you can contact CIRM to discuss more details. One more click. I just also want to point you to a webinar that was held a few months ago that really goes into the nitty-gritty detail of clinically compatible human pluripotent stem cell lines. And I highly recommend you viewing that if you're developing an allogeneic human pluripotent stem cell product. Next slide. I have one slide for the data sharing and management plans. Janie already nicely described the context for why CIRM is requiring our awardees to share their data and to be ready to share them by engaging in this process of developing early on a data sharing management plan and then updating it as the data actually get produced. Again, on our website, as Janie mentioned, what you can find in this location are the templates we will ask you to fill out once your award has been approved for funding. In the application itself, you have a section for the data sharing overview. Once you're funded, we will ask you to fill out these templates as a starting point for developing your data sharing and management plan. Also as a way of precaution, we're still reiterating these templates and they change over time. So always good to go back to the website to make sure you have the most up-to-date version when you're thinking about your data sharing. Next slide. Another resource, this is my last slide, is the fact that you have an opportunity on the CIRM website to look at previous review summaries. These are as a summaries actually of our reviewers, the grants working group reviewers, the comments they provided to previous applications that they reviewed. This is under the discovery section under funding opportunities. You sort of have to navigate down on the web page where you can see a link to these summaries in one more click. When you click there, you'll have a drop-down menu for the different cycles we've had for DISC awards, and you can start browsing through these review summaries. All right, next slide. I'll hand it over to Chan to talk about common issues and lessons learned.
Great, thanks, Uta. This is just for us to wind down as a way to summarize all the information that we've just shared with you, to kind of highlight some of the common issues that we see regarding applications that come in. So some common issues that are affecting eligibility. So you just have to make sure that you are including key activities that we expect you to need to meet the expected deliverable. Make sure that you're selecting the correct track for applications. Another segment of not being eligible is failure to include the requisite testing with human cells or human stem cells, or including out of scope or stage inappropriate activities. Just to remind you that this too is not a foundational or basic biology award, nor is it a sort of later translational or developmental award. So it's somewhere in between, and you should familiarize yourself with the requirements in this specific application. And then another category for lack of eligibility is lacking a basis or rationale for using stem cells or genetic therapy. A common one is not using the appropriate template. So these templates for the proposals are part of that application website. So you should download them and use them and not use your own template. And of course, incomplete applications and where applications have less than 20% PI effort. And the next slide. And this slide summarizes some of the common issues that affect the merits. So whether or not it's likely to be granted the award. And as Uta mentioned, a very good resource is to go to the website and look for the summary of previous review scores and learn a little bit about what kind of applications get funded and what do not. So some of the common issues, I'm going to read them here, is applications that are unlikely to meet the expected outcome within the time frame. If your preliminary data is insufficient or unconvincing, if you have weak alignment with some priorities, so a very superficial link to stem cells, for example, or regenerative medicine. If your application has high technical risks without sort of showing how you are trying to address those, missing expertise on the team, especially for areas with high risk. For grantsmanship, failure to acknowledge existing literature, for DEI statement are also factors that could come into this. So finally, on the last slide, these are just some of the, we're going to end here, and another reminder that the applications are due May 2nd, 2 p.m. PDT, and these are some of the emails you can contact us at. Primarily, you can get to the team by using discovery@CERM.ca.gov. So I'll hand it back to, I think, Janie, who's going to walk us through the Q&A.
Yes, thanks, Chan. So now we'll answer any questions that you may have. Feel free to deposit your questions in the Q&A module that's located at the bottom of the Zoom screen. I see that a few of you have already filled out questions, which is great. We'll get to those in a second. And a few have been deposited when you registered for the webinar as well. So we'll start with one of those. And the first question is, can other academic institutions outside of California be included as collaborators? And there's a related question of, what's the policy for a foreign collaborator? Can I list a foreign PI as a co-PI or a budget subcontract? So I'll send this to Uta.
Sure. The simple answer is yes. You can include out-of-state collaborators, domestic or foreign, and they can be paid through subcontracts for activities for your project. The one important piece of information to remember when you consider that is just to be clear about this and talk with your collaborators about this, is that any new intellectual property that gets generated during the CIRM award, as part of the CIRM award, must be retained by the California-based grantee.
Excellent. And we have a question. Would the DISC2 program fund a therapeutic candidate, stem cell based, in combination with another drug? Kelly?
Yes, that would be a combination product. So since on my first slide, when I described proof of concept, you want to make sure that your therapeutic candidate, it's going to include a cell and some other component. So whatever that final combination is expected to be, that's what you need to demonstrate your proof of concept with. The allogeneic versus autologous consideration is going to apply to the cell part of your therapeutic. And if your combination product includes a small molecule, or, depending on what the other components are, you should be thinking long-term about how you're going to translate that. The question might not be as important right now, but, if your component includes something that you have to acquire from a vendor and that's your only source, you might think about whether or not that is something you might need to replace or test a different component during your discovery award so that you can have something you can translate at the end. But the short answer is yes, you can test cell therapy combination products as long as the cell therapy component meets the stem cell requirements that I outlined.
Awesome. And Ute, for the demonstration of disease-modifying activity, does this have to be in an animal model, or can it be in an iPSC model?
Yeah, I'm unmuted. Demonstration of disease-modifying activity can be obtained in both an animal model and a human iPS cell-based model. Typically, and Kelly, chime in if you want to add additional information. Typically, what we see is that the disease-modifying activity is obtained in a small animal model appropriate for a DISC2 award that is considered a good disease model for the disease, and an induced pluripotent stem cell-based model would be an additional piece of supporting information for disease-modifying activity, and as a matter of fact, as Kelly explained, required to show that your therapeutic works in human cells. Kelly, did you want to add anything to that?
Yeah, I'll just add that it's up to you to define what the relevance of the model that you choose to show the evidence of disease-modifying activity for your cell therapy candidate and or other candidates. And an in vitro model is acceptable if you can provide an argument that that is providing evidence of disease-modifying activity. And as Ute said, we will often see a combination of in vitro and in vivo models proposed. There have been situations where there is no good animal model or no appropriate animal model, and an in vitro model has been proposed as an alternative way of demonstrating that. And so that's, it's the reviewers, of course, will determine whether or not they're convinced by this, but definitely it's allowable.
And I think what you said, Kelly, was the key. Is up to the grants working group to review the state of the science. There are very poor models for certain diseases, especially neuro diseases. So if you can have two models, better than one, probably an animal and a cell-based model.
Awesome. And Rosa, could you speak about how the development of new programs, including the CIRM neurostrategy, will affect the priorities and scope of the discovery programs?
Thank you, Janie. So as you all know, CIRM's new strategy was started implementation in January 2022. Since then, we've been aligning the different programs, but we are not done. So that's why you keep seeing changes. There was a question about the DISC-0 foundational program. When would be the next one? So just as a glimpse of what's coming, one of the first things that our team is focusing on right now is the neuro strategy implementation within the DISC programs. We are developing a new concept that we hope will be coming to the board of CERN for approval later this year as fruit of the Neuro Task Force recommendations with a focus on neuropsychiatric disorders. It's not approved, so it's not a done deal yet. And we are thinking about a concept that would involve multidisciplinary teams leading to some sort of a consortia. And this could also align with our data infrastructure. So that's one concept, and it will be focused on foundational type of work. So if you are thinking foundational and you're asking about D0, but your expertise is in neuropsychiatry, that's going to be covered by this new concept. With regards to the other programs, we are going to be developing very likely a new program for within DISC that will focus on biomarkers. But that's what we are thinking in terms of coming towards the next round of concepts.
Excellent. And this is a common question. How do you define a stem or progenitor cell, or does my particular stem cell qualify for funding by CERN? Kelly, you can take this one.
Yes. So we'd ask that you please review the definition of stem cells and the definition for progenitor cells that are provided as footnotes in the DIS2 program announcement. These came from the proposition. And if you can provide an argument that the cell that you're working on meets this definition, or if it's already been largely established in the field that this cell qualifies as a stem or progenitor cell, then certainly it can be an eligible subject of study in a DIS2 award. If you're working with a cell that you maybe discovered that you believe is a new type of stem or progenitor cell, but maybe the reviewers, if you have any concerns that the reviewers might not agree, be sure to provide the justification for the evidence for how that cell meets these definitions, and that would be a way to address that.
Can the application have a co-PI or co-I? And if not, can the co-PI or can the PI include application letters of support from collaborators or consultants and institute directors? Chan?
Yeah, I think you can have a co-PI. The grant will be awarded to one primary PI who's going to be in charge of the overall ownership of the project. But you can have co-PIs and other types of key personnel that's listed in your application to provide the requisite knowledge base to get a successful grant.
Maybe I could just elaborate that on one point, since we've heard that some applicants consider co-principal investigator somebody who would be managing their own part of the grant budget. That type of arrangement is not one that's supported in DIS2. DIS2 grants identify one individual as the principal investigator, and all others are technically classified as key personnel. However, you are allowed to define the roles of each key personnel, and you can define that role as a co-PI or as a co-investigator, as Chan said, and you would define what their responsibilities are.
Do you have to have any publications for the DISC grant, or how much preliminary data is needed for these awards? Hallie?
I can try to take that one. So there's no one size fits all formula for how much preliminary data is required, because it's going to be different for every type of project, and how much of the rationale might have already been established by pre-existing literature, or how much of it is completely new. The important thing is that reviewers do look at preliminary data because they use it to assess the overall risk of the project, both in terms of whether the rationale makes sense, right? But also whether or not it's going to be technically feasible to achieve the project aims and identify a development candidate that will be ready for translation at the end of the project period. So if you're really early along and there's a lot of technical risk and it's a very complex project, it might appear a little bit less feasible to them if you don't have very much preliminary data. On the other hand, if you come in and you have compelling preliminary data, published or not, that shows that you can do these techniques, that it's in your wheelhouse, that you have, you know, strong support for the rationale, and they believe you can do it, then you know, that might be good enough. So as I said, it's not a one size fits all formula, but it is very important to help reviewers assess the merit of your proposal.
Perfect. Can out-of-state collaborators get salary support or only support for lab supplies? Uta?
The support that goes out through a subcontract, there are no rules related to these budget line items, and CIRM doesn't review that. So both salary and supplies can go to out-of-state collaborators in a subcontract.
How critical is it to show solvency for for-profit enterprises? How long of a cash run is sufficient for the purposes of this grant? Kelly, do you want to take that one?
Okay, you need to demonstrate 180 days cash on hand at the time of application. So that will be, in order to be reviewed, the application materials will, our review team will be looking at that to ensure that for-profit applicants meet that solvency requirement at the time of application. Oh, I should also mention that if your grant application proposes any type of co-funding or is bringing anything else to the table beyond the DISC funds that would be offered by CIRM in order to achieve the project outcome, you'd also need to be able to demonstrate the ability to make those financial commitments.
Okay. And are project managers required, and what is their % effort that's required? Uta?
There's no requirement for a project manager in the DIS2 program at this time. It might not be a bad idea to have one if it's a complex project and there's a lot of tasks, but it's not a requirement in this program.
And Kelly, if I can chime in, however, for your data sharing and managing data managing purposes of your project, actually in DISC2, so we highly recommend that there is somebody dedicated to the data sharing and managing part of your proposal. I got to explain, I got confused. In DISC0, actually, we did require that there be a data manager, and in the future that might happen in DISC2 as well.
Great. And Uta, can you also share how to address diversity, equity, and inclusion in project applications?
Sure. Addressing diversity, equity, and inclusion, is an important part of the review process. And my first comment is for you to pay good attention. You have a whole section in your proposal template that's dedicated to diversity, equity, and inclusion. And it actually nicely outlines 3 subsections with examples and explanations. So I think that should guide you quite well through this part of your proposal. I also recommend that you go back to the program announcement and look at the review criteria, so you have an understanding of how the reviewers are instructed to assess the quality of your DEI section. And then I also actually would like to point you to an FAQ document we have on our website and the discovery part for funding opportunities. Under the DISC0 portion, there is an FAQ document that addresses in a little bit more detail some of the suggestions or considerations that CIRM is proposing you consider. And so briefly, I think it's, and having listened, having listened to how these reviews go, it's very important for you to show that you are specific and intentional about your diversity, equity, and inclusion approach. And what I mean by that is that you shouldn't say, yes, I will use many diverse human pluripotent stem cell lines, for instance. Instead, you should be specific and say, for the reasons outlined in my proposal, these specific types of stem cell lines, would address diversity in my research project, be it gender or sex diversity or ethnic diversity, et cetera, or ancestral diversity. Explain why you're choosing very specific certain ancestry as depending on your project. With intentional, what I mean by that is, again, that you shouldn't just say, I will include the diversity of our patient pool for recruitment, for instance, for obtaining cells. Instead, you should say, this is my approach to making sure that I will be able to reach those diverse populations. So be intentional when you describe your diversity approaches. And the last thing maybe, and these were just two examples I gave. This is, of course, a much broader subject. But it's also, reviewers will also look at whether you understand what the diversity issues and inequities are that are related to your specific project. Do you understand what they are and what you're proposing to address them? Is it likely to address these inequities? And if you don't yourself have that knowledge, you can always, as CIRM funding actually does allow for you to have a team member who can help you understand that better. Or you can also delve into institutional resources that your institution might have. And again, be specific and intentional when you address your diversity, equity, and inclusion approaches.
Kelly, can the iPSCs from CIRM be used without licenses by California companies?
You are allowed to acquire cells from the iPSC repository and use them without any kind of special license for the research in your laboratory. Is there anything else you might want to add to that, Uta? Or?
Yeah, we highly welcome you to make use of the CIRM iPS repository. Lots of, there are limited diseases addressed, but there are lots of choices per disease. There's lots of diversity in ancestral diversity in this bank. So we do highly recommend to use those cell lines for research.
Yes, and just to hammer the point home, those cells are not appropriate to use as your therapeutic candidate, as your final therapeutic candidate, if it's going to be an allogeneic therapy.
How is confidentiality and IP maintained by CIRM during this process, especially for pre-DC'd structures? Should a composition of matter patent be filed in advance of submission, or are such structures not required?
Well, that would be a good question for someone from our review office who I know are present in the audience. And perhaps if you put that question in the Q&A, they can give you a more specific answer. We're maintained by confidentiality as employees of CIRM, and I know that our grants working group members all maintain that as well. But the actual specifics for how they apply that is a question for our review office, and we'll make sure that somebody provides an answer for you if you leave your question in the Q&A.
Fantastic. We'll leave that up there. And if we use the chimeric antigen receptor, or CAR, in a stem cell, and the stem cell aspect is completely novel, does the CAR need to be novel or protected by institutional IP, or can we use an existing / generic CAR?
Can you say it again?
Yes. So they're proposing using a generic chimeric antigen receptor in a CAR in a stem cell, and the stem cell aspect is completely novel. But they're asking, does the CAR need to be novel and protected by institutional IP, or can they use an existing CAR?
You can use an existing CAR, you can use a novel CAR, you can use a novel stem cell, you can use an existing stem cell. Presumably what you propose to discover is going to be a novel entity when you take the whole thing together. So yes, that's fine.
Mm-hmm. And this person is asking a clarifying question. A gene therapy developed to reprogram the brain cancer stem cells as a therapy is eligible for TRAN1. Is that correct?
Well, we're talking about DISC2 today, not TRAN1. So I would defer that question to members of our therapeutics team who manage that program. But we could always have a conversation about that in the context of DISC2 and TRAN1 if you would like to contact us through the discovery program, because we can help you consider whether what you are proposing is actually discovery stage or translational stage and how to fit it into the most appropriate funding opportunity if it's an eligible candidate.
Okay, and now they are actually, there's been an edit that says they meant DISC2.
Oh, okay. So reprogramming, did you say glioblastoma stem cells in the brain or reprogramming brain cell?
Reprogramming brain cancer stem cells as a therapy.
Okay, well, if you can make an argument that it's a stem cell and it's a target of your therapy, then that could potentially fall into one of our eligible categories. If you provide these arguments and put together a responsive proposal.
But maybe I can also add, if the reprogramming requires a nucleic acid, you could also make an argument that this is a genetic therapy. And you can certainly make both arguments in order to really demonstrate that this is very much in scope with what CIRM is interested in, but genetic therapy is another option.
Okay. And just a note about resubmissions from other tracks. Can you resubmit a DISC0 application in a DISC2 application, Kelly?
That would not be advisable in general because, well, there may be some cases where a subset of the proposed activities that you're doing could be similar between the awards. The overall objective and deliverable for the program is quite different. And so the reviewers are going to be looking at them from a different perspective. So there may be some exceptions to this, but for the most part, it's generally not advisable. And if you would consider that you might fall into one of these exceptions, you might want to reach out to us to discuss that before you try to repurpose an application.
Do you use outside reviewers that could be competitors, Uta or Chan?
Well, maybe I'll
they can send that to, they can put the question and the review will answer. But no, we go through a very thorough conflicts of interest process. So, and the answer straight is no. And you can ask and the review office will provide you the long answer. Thanks.
Excellent.
Also, I just wanted to mention that we have a consultation form. So if somebody wants to repurpose a DISC0 for a DISC2, please fill up. There is a link and there's an auto response right now in the discovery inbox, and you can complete the link that will submit a form. And we want the questions, the specific questions. Obviously, the goal, as Kelly was saying, is very different for the DISC2 because the DISC2 is the entering to the pipeline of development towards a successful therapeutic candidate, whereas DISC0 is foundational work. So there should be some changes. So try to think what those changes are and provide suggestions so we can help you and advise.
Great. And this question is about evaluating clinical translatability of DISC2 projects. What is the matrix that is used to evaluate whether something is clinically translatable at the end of your DISC2 award?
You know, I'm not exactly sure what this question is asking, but if you're asking how is it determined whether or not the program would be translatable at the end of a DISC2 award, eligible to move into further stages of development, we use our definitions of readiness that are based on the deliverables that I described. So you're going to want to have the candidate defined, and you're going to want to have for sure evidence of disease or injury modifying activity with that candidate. And you're going to, a draft target product profile is a good idea. Having preliminary measures or characterization of your candidate is a good idea. If you manage to meet all these deliverables, you should be, for most eligible candidate types, eligible to apply for follow-on funding through our translation stage program. You can always look at the translation stage program announcements for what the entry criteria for that program is, because that's where you're aiming to go at the conclusion of your DIS2 award. If everything works out perfectly, we would love to be able to support the candidate through continued development if it's at the right stage to be competitive for the Tran 1 proposal. The other important consideration I mentioned is, if you're developing a allogeneic cell therapy candidate, you're going to want to make sure that your candidate is something the FDA is going to let you take forward. If they are derived from a donor that was not given the appropriate testing at the time that they donated the cells to make those pluripotent stem cells, they're not going to be able to be used in a clinical trial. So that's why we referred you to some resources so you can learn more about clinically compatible cell lines. And so you can use those in your studies from the get-go and not have to go back and change cell lines and start over in order to demonstrate your disease modifying activity.
Yeah, but Kelly, the TPP is the last milestone, is a milestone that we want to see, and these two. And the entry criteria for Tran 1 are very informational because that's where we want to do. So those are the two main references that they should pick.
Now, one thing we didn't go over much in the webinar today, because this was really focused on helping you put together competitive applications. If you are awarded DIS2, we will work with you to help you develop strengthen your TPP. We will do whatever we can to help you be successful, because the goal of this program is to produce development candidates that can and will be developed. And so we're doing everything we can to help you succeed in that endeavor so that you'll be able to secure follow-on funding at some point to take it to the next level. I think we're getting a little tight on time now. We've gone over a bit, and we want to be respectful of people's time. So we have answered a number of additional questions in the Q&A, and we will make efforts to address the rest of them, and we'll write up responses to those, and we'll make that available to everyone along with the recording, and we'll make the slides available as well. So thank you very much for joining us today. We look forward to talking with you further. If you have projects that you're interested in submitting, reach out to those contact emails that we mentioned. They're also in the program announcement. And we look forward to successful applications and potentially working with you in the future.
Bye.